Bats are a common source of zoonotic viral infection, many of which are well known even to non-scientists. They are a source of rabies, a persistent scourge to the human population, as well as Ebola and Marburg, terrifying diseases that strike fear into society. They are also a possible reservoir of SARS-CoV-2, a virus which has impacted all of us. However, they are also the host of a family of less well-known but terrifying viruses, the Nipah viruses.
In 1994, Brisbane, Australia saw an outbreak involving 21 horses and 2 humans. The infected horses and humans developed severe respiratory symptoms of an unknown disease. One of the humans died, and of the 14, unfortunately, the horses had to be put down. This new virus named Hendra virus was isolated and sequenced. The virus is recognized as part of the Paramyxoviridae family, but the exceptionally large genome compared to other viruses in this family led to the virus being assigned to a new genus, and will later be joined by another emerging virus in Malaysia.
In 1989, a mysterious outbreak struck Malaysia with 105 out of 265 cases, resulting in death. Neurological symptoms meant that it was initially assumed to be Japanese encephalitis virus. However, efforts to vaccinate the population against Japanese encephalitis were not effective, and cases were associated with pig farming, which is not a characteristic of a mosquito-borne Japanese encephalitis. Immunological assays found a similarity to Hendra, but gene-level sequence differences of around 21% indicated that it was a closely related but distinct virus. Eventually, the epidemic was halted by taking multiple measures, including culling the pig population in the affected area. In 2001, a similar outbreak struck Bangladesh, leading to 13 cases. Since then, outbreaks have frequently occurred and have led to 199 deaths out of 261 cases. Unlike Malaysia, these cases were primarily due to consumption of date palm sap that had been contaminated by droppings and urine from infected bats. As demonstrated by the details of these outbreaks, Nipah and Hendra viruses differ in 3 key areas: the symptoms produced, their geographic location, and the means by which the virus jumps from the bat population to humans. Hendra virus involves infection of horses and is then passed from horses to humans, whereas Nipah is primarily spread to humans either by infection of pigs or by contamination of date palm sap by bats, which is then consumed by humans. Hendra is found in Australia, while Nipah is found in Southeast Asia and India. Lastly, the symptoms of Hendra are primarily respiratory, while Nipah has multiple neurological symptoms.
Despite these differences, Nipah and Hendra share common molecular level features. Both are negative-sense single-stranded RNA viruses. Their genomes are bound by their nucleocapsid protein N to form an RNP, which also contains their phosphoprotein P and polymerase protein L. They are envelope viruses containing 2 spike proteins, glycoprotein G and fusion protein F. The G protein binds efferent B2 and B3, which are highly conserved receptor tyrosine kinases and exploits the cellular macropinocytosis pathway. Conservation of an efferent B2 may explain the broad host range of Nipah viruses compared to other paramyxoviruses. Another aspect of these viruses that separates them from other paramyxoviruses is their genome size. They are both around 18 kilobases, approximately 2,700 nucleotides longer than others in the family. It's been found that these extra nucleotides form large UTRs.
A key feature of the pathogenesis of both viruses is their ability to evade the innate immune system. The V protein prevents STAT-induced activation of interferon, allowing the virus to evade the early immune response. This is a feature that is generally conserved among paramyxoviruses. Interestingly, Cedar virus, which is closely related to Hendra and Nipah, does not produce a V protein and does not result in disease in humans.
For both Nipah and Hendra viruses, the emergence of these viruses can be attributed to increased human encroachment into the habitat of fruit bats, which are the natural reservoir of these viruses. When pig farms or horse pastures were built in areas where these fruit bats resided, Nipah and Hendra virus are able to spread to pigs or horses through exposure to bat droppings, secretions, or even half-eaten fruit. Once the pigs and horses are infected, the virus can be transmitted to humans. For Nipah virus, pig-to-human transmission can occur via urine, saliva, or respiratory pathways. For Hendra virus, horse-to-human transmission can occur via exposure to blood or secretions of infected horses. In the case of Hendra virus, this is rare. Only 7 humans, 4 of whom did not survive, have been known to be infected, and no human-to-human transmission has been documented, displaying an example of dead-end transmission. Primarily, horses are infected, with several notable breakouts among groups of horses. Hendra virus then occasionally makes a jump to humans that were in close contact with the infected horses. Human-to-human transmission of Nipah has occurred, although is not sufficient to sustain transmission.
The diverse mechanisms of zoonotic transmission provide multiple lessons about managing such animal-borne diseases. One approach to limiting Nipah spread has been addressing environmental factors that contribute to crossover. For instance, in Malaysia, keeping fruit trees, and thus bat roosts, separated from pig farms is one mechanism that has been explored to reduce transmission rates. This addresses the overlapping anthropogenic and ecological factors that contribute to transmission. It demonstrates that sometimes diseases can be addressed without using therapeutics or vaccines. Another strategy has been vaccinating the intermediary species in Hendra transmission, horses. Developing and distributing a human vaccine would perhaps be impractical, considering the low number of human cases and lack of human-to-human cases. However, a vaccine for horses protects a species that is at a far greater risk of infection, and in doing so, prevents crossover from horses to humans. This demonstrates the benefits of a One Health approach in which human and animal health are viewed as interconnected.
Another lesson comes from how the Nipah and Hendra viruses emphasize the role of bats as reservoirs for emerging viruses. Another emerging virus that has been linked to bats includes SARS-CoV-2, Ebola, MERS, and Marburg virus. It is important that we learn about the range of bat species and identify the ways that humans and animals interact with bats so that viral spread from bats can be limited. As climate change leads to changing ranges of host species, and as humans continue to expand into these ranges, the emergence of viruses like Nipah and Hendra virus will continue to increase. Especially because these viruses have extremely high mortality rates, it is crucial that we use our knowledge in virology to identify, contain, and control any new or re-emerging viruses.